All right, folks, welcome to the second lecture of week 8, where we'll be talking about all the fungi, including the mushrooms. Caption continues to be a problem, and so we'll see what we end up using. We might be going with Zoom caption right now because it is, in fact, able to record the transcript right now for me, even though you guys cannot see it. All right, general announcement. So to remind everyone again, we have on week 10 2 exams that week. On Tuesday of week 10 is your second lab exam, where we'll be covering materials on week 4 through 8. That is our main focus. But remember, for both the lab exam and the lecture exam, there is a cumulative portion where 25% of the question will be based on what you guys missed from the previous exams. October 30th, Thursday of that week, is your third lecture exam, and this is covering or focusing on the material from week 7 through 9. And yes, there is a cumulative part for that as well, which will comprise up to 25% of the total score. Again, the day of your lecture exam is the day your review assignment is due. Number first is a point race field trip. For more details, it will cover that in the lab intro slides. November 11th, no lab, no class. November 18th is the drop deadline for the W grade. And November 20th will be our lab exam number 3. Now let's go directly to the knowledge review. So name one trait that distinguishes amoeba from algae. And there are many, many things you can put here. Go ahead and pause the video and then we'll go over the answer. So there are several things you can say for this, but I hope that the key one you guys will remember is that amoeba are heterotrophs and algae are photoautotrophs. So amoeba needs to eat things to live. And that is actually what ties in really well with the algae, because algae are also heterotrophs. So that brings up to today, which is our algae lecture. So we're going to introduce and orient ourselves to the algae, and then we're going to talk about 3 of the main families in there. So algae are eukaryotic heterotrophs, so they do not do photosynthesis. To obtain nutrient this way, they have to excrete enzyme to digest things, and then they absorb what's digested. And this requires water. Here on the right is a nice illustration of the importance of the symbiotic relationship between plants and fungi. So in the small photo on the right, you can see this is the root size of this gymnosperm. Usually the root size by itself is at least the same size as the shoot, and it can be bigger, but it's not big to the same stage as you see in the photo on the left here, which is another gymnosperm. I think this one is a pine. And remember, one of the key importance of mycorrhiza, that is the structure of the symbiotic relation between fungi and plants, is that it can extensively expand the amount of areas the plant can explore with its roots, because now the fungal hyphae is acting like an extension of the root, which can then cover a lot more area. So there are several types of heterotrophs. There are saprobes, which are the decomposers. That's what we tend to call them. And they feed off dead organic material. There are, however, parasites that live off of living organisms, so absorbing nutrient directly from live organisms. And they are mutualist fungi where they live off of living organism. Usually they require them for support, but they are not harming or providing benefits to their host. Parasites harm the host. And then, of course, we have symbiotic relationships where both sides, the host and the symbiote, will benefit. So on the left here, we have a decomposer helping break down the dead trees. And then on the right, you have one of the many types of disease that fungi can cause in the plants. So plant-fungal relationship is not always symbiotic. It can be pest and pathogen versus host response, especially if you guys remember the PEM and MEM from our plant immunity. There are a lot of fungi that can cause diseases in plants, and they are classified as pathogens. The fungi do have cell wall. So in this case, unlike the various algae and Producer-Carabot, the cell walls of fungi is made of chitin, which is the same material that makes up the exoskeleton of insects, shown in this middle image on the right. Fungi reproduce by spores. So this is what this Ascomyces on the left is doing, is releasing its spore. And they can reproduce asexually or sexually. You can have spores either way. They do zygotic meiosis for your life cycle. So if you recall from the end of our last lecture, zygotic meiosis means that the meiotic step is occurring right after the formation of the zygote. So in this case, it is the haploid form that is dominant. Here's what chitin looks like. So you do have a sugar molecule there on the top. And one of the things that is economically important about chitin is you can use it to make really good surgical thread. But I'm sure you guys know there are many other reasons why fungi are important, right? Because as decomposer, it is a key part of the ecosystem. It is important for the mobility of the nutrient cycle. By breaking down the dead organic material, a lot of the nutrients are thus re-released from where it's locked up in the body of the dead organism, and then those nutrients become available to the other organisms in the ecosystem. As such, fungi is a key part for both nitrogen and calcium geochemical cycles. So we can see basically here is one key part, decomposition by fungi and bacteria, and fungi do tend to be a little bit closer to the surface of the soil compared to some of the bacteria. The other important role for fungi is for the study of evolution and diversity. Fungi are some of the largest and oldest organisms alive. So there is a appropriately named fungus called the humongous fungus. It is over 2,000 acres and 8,000 years old. And when we cover the hyphae, you can see how they can get so big. But this is how they, okay, we'll cover that later in a later slide. And we just covered some of the beneficial reasons why they're important to study, right? Here's a not so beneficial reason of why we should study them. And that is all the disease they can cause in both animals, including us, and in plants. But they are also the source of medicine and food. I'm sure everyone has heard of penicillin, right? So penicillin is an antibiotic. They can kill off bacteria. That's what antibiotic means. It kills off bacteria and some microbes specifically. And I do want to mention that for this reason, taking antibiotic when you have a cold or flu does not work, because cold and flu are caused by viruses, and antibiotic works not on viruses. they work on bacteria. So you need to take antivirals for any disease that's caused by a virus. But anyway, here on the left, you can see the circle around the penicillium colony where you have inhibition of bacterial growth. And if you guys are curious, you can look up this project called Tiny Earth, which is very similar to our Cure project, except instead of sampling trees, they sample soil from around their neighborhood, around their campus, and screen the soil content for things that show antibiotic property. So that's very cool. There are many schools and campuses involved. Everyone is, of course, familiar with the mushroom we eat. There are many kinds of that. And morel and truffle are all fungi. But fungi is also important for biofuel as well. Specifically, they contain cellulase, which can break down and digest cellulose to release glucose, which then can be converted to ethanol as biofuel. And if you guys recall from week 2, cellulose is one of the most abundant biopolymers in the world. However, breaking it down can take energy, and if the amount of energy you put into breaking down cellulose is greater than the energy you can get from the biofuel, then that fails the endeavor because you are putting in more energy to create an energy source than the energy you're getting out. So it's no longer sustainable. So people are always looking for new way and more sustainable ways of breaking down material to make biofuel. And cellulase is something that people are using because, well, it comes from fungi. So it is eco-friendly and more sustainable, right, than heating something and treating it with acid. Another thing that's really cool, and this is a pretty hot topic in the recent years, is the use of and creation of sustainable material from all the material that exists in nature around us. And some folks have found out that you can use mushroom to create different kind of building and clothing materials. So you can create mushroom-based cork and concrete and leather. And so that means takes us to the orientation of fungi itself. So fungi can be unicellular or multicellular. We know yeast that tends to be fungi that grows in unicellular form. So that's the top one over here. But a lot of the fungus we see around us that we're familiar with are multicellular. And the specific term for their multicellular filament form is hyphae. And when you have a bunch of those filaments together, it's called a mycelium. So the plural form of mycelium is mycelia. And the hyphae can be aseptate or septate. So aseptate is co-inosittic, so that means you do not have cell division. So if you recall back to the last lecture when we're talking about slime modes, right? And for the plasmodium form or the feeding form, when the slime mode does division, the cells do not do cytokinesis. The nuclei keeps dividing, so you end up with just giant cell that's multinucleate, but do not have cell walls. And I mentioned then that the fungi does something very similar. Well, here it is. It is the aseptate form of fungi, where mitosis for the division only result in the division of the nucleus, but there's no cell wall separating each of these. And so that the cell wall that divides the hyphae is called septum. There's no septum, so it is aseptate. The opposing end of that, of course, are the septate hyphae. So each time when the cell divides, the nucleus divides, a new cell wall is formed. And so the cells are now separated by septa. The singular form for that is septum, plural form is septa. And the septum tend to have pores. what's illustrated here. Here is what that will look like under the microscope. So on the left, you can see the hyphae with their nice septa. And on the right, here is a septa showing the pore in the middle. All right, now for quick knowledge check. The two main types of fungal hyphae are, or they can be divided into, which two categories, depending on the presence or absence of septum. Go ahead and pause the video, and then we'll go over the answer.